Link for the quiz is given in the comment section. Everyone, please join using the link given in the comment section. Also, we'll share the link. We have shared the link in Telegram and WhatsApp groups. Okay.
Hi guys, I hope you're prepared for today's topic. Today's topic is genetics. It would be a small session and we'll be starting the quiz exactly at 10:10 p.m.
We'll be starting in exactly 4 minutes. Okay. We'll start the quiz exactly at 10:10. We have total 19 candidates who are ready. I think many will join as we reach 10:10. Or is it that everyone is everyone is busy because it is Sunday and you want to rest?
Might be, sir. And guys, even if you have not prepared for the quiz, it's okay to attend it.
Yeah, because we have covered all PYQs, PYTs, so it will be revision for you. Okay. So what are the questions that are asked? Similar questions only will be coming in exam also. So in one or the other way, it will be like revision for you. I think
Yes.
students have realized that quiz will start at 10 p.m. So yesterday I noticed that between 10:08 to 10:10, like there were 50, 60 entries. Till then, there were only
Yes,
10
sir.
to 20.
Yeah. They're managing their time efficiently, sir.
Yeah. So we can see previous winners in the players who have already joined. We have Sriraj, Hemant, Kirtana, there is Jaspreet, there is Soumya,
Ashish, Tanya also. Hemant.
yeah, Ashish and Tanya. Good that more and more people are joining. So
Hmm.
today's topic is genetics, one of the very important topics for INICET. Even in last INICET, many questions were asked from genetics. Okay, so INICET, if you see last few years trend, many questions are focused on on genetic. That is because INIs, that is aims, is giving more importance to genetic workup. Okay, and this DNA analysis, syndromes from DNA. Okay, they have separate genetics department. I got the opportunity to attend their CME and interact with the faculty over there. So I realized that they have done many advances. Okay, so when these faculty from these institutes start preparing questions, they will be uh, asking the same questions in which their research is going on. And research is going on
Yes,
in
sir.
aims regarding, especially regarding mucopolysaccharidosis, (MPS). Okay, new genetic signal, the whole exome genome sequencing. All these are like hot topics for research in aims. So
Oh,
that's why they are more focused on these questions. And when you note also this, the questions that are asked now are more based on enzyme replacement therapies. They're more based on genetic approaches.
hmm. Yes, sir.
We'll be starting in exactly 2 minutes. Okay. I guess many are enjoying their Sundays. I think that seems to be the reason, Manoj.
Hmm. Yes, sir. Might be. They're not taking Sunday seriously for studies.
Even one day will make a difference now once that your preparation has started. So
Yes, sir.
those who are like consistent enough studying every day, they will be the ones who will actually win this race.
Yes, sir.
Mhm.
True. True. True.
Okay, we have 1 more minute to go. We'll start within 1 minute. We are always we always follow time. We are very punctual. Okay, so let's start. Welcome
Let's go.
everyone to the JuxtaMed quiz. I'm Dr. RMD, your quiz master for today. This time we are diving deep into the world of genetics, from the smallest mutation to the most intricate genetic disorders. We'll be testing your knowledge. With me are my co-hosts, Dr. Manoj and Dr. Rupal, who will be helping out with the clues to guide you. Let's see who's ready to take on the genetic code.
Mhm. Let's go.
Here is your first question.
A 2-year-old boy presents with delayed developmental milestones and self-mutilating behavior, including biting his lips and fingers. So lab investigation revealed hyperuricemia, which enzyme deficiency is most likely responsible for this condition.
So, in case you guys are looking for a clue, here we go. This enzyme deficiency is often associated with a genetic disorder where the affected individual may have neurological defects and hyperuricemia. It's one of the purine metabolism disorders.
So the clue self-mutilating behavior and hyperuricemia should guide you to diagnosis. So most of them have got it right. It's frequently asked PYQ. So I don't think there is any mention of need to mention anything else. So let's see at the leaderboard. Okay, we have Krishna who is leading right now, followed by Soumya, Meera, Malcolm who are at the same position, followed by Sriraj.
So,
Let's
can
move
you
on to the
see? Three people are on the same position. The competition is so cutthroat. Oof,
yeah, right.
since the
It's cut
start.
pharynx, I guess, not even cut.
Okay, so second question. A female patient presents with short stature, webbed neck, and primary amenorrhea. Karyotyping reveals a 45XO genotype. Which of the following condition is most likely? I
So this is a classical chromosomal condition. So there's no doubt the image itself is enough for diagnosis.
that's true.
Yes, sir.
That's true. I think these genetic disorders should be on everyone's tip.
I don't know why few
Yeah.
of them were so much confused. I think if the clue was Noonan syndrome, then students would have been confused a bit.
Hmm.
Let's focus towards the leaderboard. It's Soumya leading now and Malcolm both, followed by Karthikeyan, Jasprit, and Ashish. Good going, guys.
All right.
Here is your third question for the day. Which gene mutation is responsible for Marfan syndrome? This is
So
also
to give
this
a
often
yeah.
comes as a match the following question.
Hmm.
Hmm.
Yes. And
So
in
to give a
case,
clue, this
yeah,
gene is located on chromosome 15 and it codes for a protein that is essential in connective tissue structure.
it's known for causing issues in the heart, eyes, and bones. And if you still don't get it, you'll get it. I'll tell you how. Fibrin 1 is the gene that has been encoded in this protein. And we can see over
Hmm.
here that most of them have got it right.
Yeah. So now it's battle of fingers. The one with the fastest fingers will be at top.
Oof.
So there is huge competition right here. Karthikeyan, followed by Malcolm, Soumya, and Meera, all at same score, almost same score.
Almost.
Soumya and Meera at 279, and at fifth position we have Ashish. Such a cutthroat competition.
It is. It is.
Hmm.
All right, guys, getting up to your next question.
Which enzyme is used in PCR to synthesize new DNA strands?
This enzyme is a heat stable enzyme which is crucial for high temperature. It is also essential for elongating DNA strands from the primers. That is your first clue.
Hmm.
The
Yes.
another clue that you want, I can say it is one of the famous enzymes in molecular biology, named after a bacterium from which it was originally isolated.
When I say
I
this question, I remember a video released by a YouTube channel named Veritasium, which is a science channel. It had released a video on DNA polymerase PCR technique. So it was one of the
Hmm.
excellent video that I'd seen. I guess I'll share the link in the group so that everyone gets to watch this video. Like it was one of the excellent videos. When you explain medicine from that point of view, you don't feel like studying. You learn everything.
I think everyone is going to just click on the link as soon as you send it. Because medicine is such a big subject, and we do appreciate people or all the people, everyone who can make it easy, even if it is a one small topic.
Karthikeyan is leading
Yeah.
now, followed by Ashish, Tanya, Shashank, and Sriraj. Moving on to the next question.
Okay. The next question: Northern blotting is primarily used to detect.
Your first clue is this technique is typically used to analyze RNA, specifically looking at gene expression or specific mRNA transcripts.
Hmm. Yes. It's a laboratory technique where RNA is separated by gel electrophoresis and then transferred to a membrane for hybridization with the probe.
This northern blotting, southern blotting, western blotting, it's a very frequently asked question, especially in INICET exam. So it's very essential you know the techniques.
Agreed. Agreed.
So
Oh,
we
So
have
perfect
Ashish.
20th notebook topic, sir.
Yeah, true. So at this point of time, we have Ashish who is leading, followed very closely by Karthikeyan and Tanya. Then we have Shashank. We have Dr. Strange at fifth position, Faizi at sixth, seventh we have Kirtana, eighth we have Sriraj, and at ninth and tenth position at same score we have Ram and Medha. So
wow.
let's let's move on. This is my favorite question. Every time I comment about this, which type of DLL polymerase is used for repair? This is a pearl which has to be bookmarked.
Mm, that's
Why
right.
is it
So,
so, sir?
That is because
Did the
when I was giving JT this question, I was encountering it frequently. And it's a question which we have to memorize and we have to read it again and again to remember. And it's an easy scoring one.
Hmm.
So you have to look at this again. Before every GTA, you used to look at this pearl.
Hmm.
So this is the reason why I used to look because everyone would be confused.
I think this time everyone will make it a point to, bookmark this beautiful question.
Oh, Tanya is the one. Tanya has gained a significant lead, and she's the one with
Woohoo!
the highest streak. She's leading,
Okay.
followed by Ram, Mehta, and Meera.
All right, guys, this is very good going. This I know the competition is very cutthroat, but all of you are doing great. Each question, the faster you answer, you'll get extra points. And don't forget, the fastest person will be leading the leaderboard.
Yeah, moving on to the seventh question. If the replication fork in DNA repair mechanism is affected, what syndrome will be seen here?
And for your clue, this syndrome is caused by defect in the DNA repair mechanisms, which impacts the ability of the cell to handle oxidative stress and DNA damage.
This is one more question which I used to bookmark and read before exam. Every GT. This is because this is also one more confusing topic. You will end
Mhm.
up mismatching these options. That's why here also you'll see people have
Hmm.
done the same
Mixed
mistake.
answers.
Yeah. So that's why these two things, DNA polymerase types and this the syndromes involved with DNA mismatch repairs,
And
these two
also
are
PCR, no, sir. This frequently there,
yeah,
including PCR questions
Yeah.
we get.
right.
I think all these DNA techniques, no, they love to ask. Like one on the other way, they'll ask one of the techniques and they'll ask you what are the mechanisms and something related to it. So they love
Yeah,
asking it.
right.
Hmm.
So there's no way you can commit mistakes in this.
Right. So moving on to your next question. A mutation occurs in the Tata box of a gene, most likely to be called as TATA box. What is the most likely consequence of this mutation on transcription?
So this mutation would likely impair the binding of the transcription machinery, particularly the RNA polymerase enzyme and the other general transcription factors.
Yeah, and
Hmm.
also because the TATA box is crucial for the initiation of transcription, its mutation would cause a decrease of failure in gene expression.
Damn. So we got 23 people answering the right answer. Hmm.
Yeah, this was a straight PYQ.
Yeah.
Maiza is leading significantly and she has answered all the eight questions correctly. So she has gained a significant advantage.
Yeah.
She is leading, followed by Meera, Tanya, Ram, and Yes. So
Damn.
we see there's a huge shift toppling of the table itself.
Mhm. Good going, guys. Good going. Let's keep up the pace for your next and the second last question.
One more question from my favorite topic, PCR. What is the correct order? I think this used to be even NEET UG
Yeah, NEET
question, right?
UG question,
Yes,
classic question.
classic. And in case you guys are wondering what was the first step, it was the denaturation, where the double-stranded DNA is heated to separate into the single strand. Dr. Manoj, can you tell us the next step?
Yeah, the next step is annealing, followed by extension, where new strands of DNA are synthesized by
I guess if they
polymer.
watch that Veritasium video, no one is going to forget. That 30 minutes of that video, it was like eye-opening moment for me. I didn't know genetics could be taught in such a uh, in such a
Beautiful.
wide.
Right.
So Meda is leading again. She has not committed any mistakes. So followed by Meera, Tanya, and Ram. Last question, guys. Be quick. Which of the following mechanisms is least likely to contribute to elevated serum uric acid levels in a patient with gout? So here are your four options. Again, gout is a
I think
topic which
these,
is
yes, sir.
uh,
Uh,
which is
these
frequently
type
asked
of
in.
question decide the ranks also. I think.
Yeah, right. So all your four options are also slightly confusing if you don't study the mechanism.
Guys, this is like one of those things where geriatric things also are be contributed because as the age increases, the bone density and everything changes. So this is one of the things that everyone should keep in mind.
Let's see the final leaderboard. Yes, it's Medha who has won the quiz.
Woohoo!
So I guess she has got 10 streak. That is, she has answered all 10 out of 10.
Damn,
Wonderful,
good.
Medha.
Yeah.
You
Woohoo.
deserve it.
That is true. That is true. Congratulations, Dr. Medha.
At second position, we have Meera. Third, we have Tanya. Fourth, we have Faizi. Fifth, Ram. Sixth, Yes. Seventh, we have Ashish. Eighth, we have Karthikeyan. Ninth, Mahima. And at 10th position, we have Malcolm. Congrats, guys. So come back to
Yeah.
YouTube Live. I hope this was a good quizzing session. If you have any doubts, you can comment in the YouTube Live video section.
Guys, we would love to hear your feedbacks of any kind, however you like, and what do you want to have next. We love to hear your end as well.
Okay, Daniel has asked for more questions. More questions are available on Cubag. You can solve.
Uh,
The problem is, if it is more questions, students will lose interest after 10 questions. After 10, 15, they'll be like, okay, this is nothing different than QBank.
Cuba,
So that's
yeah,
why we have broken down the whole quiz into small topics.
that's true. That's true. And guys, keeping the session a little, small and sweet is because it makes it a better. It gives you a little feeling of a new thing, a fun thing, and then over here when you feel rewarded, also, it just adds up to the whole blend, and you feel validated. So we're just trying to keep you in touch with your studies, also, along with that, having a flow and consistency towards your future goal.
Yeah.
Someone has asked for that PCR video, which I was discussing. Yes, I'll send the link of the PCR video. Okay, it's a very good video. Like it was an eye-opening moment for me. I didn't know medicine can be explained in such a beautiful way. Okay, I'll send the link in the group soon. Thank you,
Wonderful.
Harsh. Harsh has thanked us for these sessions because they are really good.
Oh, thank you. Thank you.
Thank you, guys. Thank you.
Okay, so tomorrow we'll be having proteins. Okay, so focus on proteins, especially amino acids that are very important, like tyrosine, which is very important. We have phenylalanine, which is very important. We have branched branch chain amino acids in that, especially disorders like maple syrup urine disease, which are very important. We have some urine orders, okay, which are very frequently asked. Were asked earlier. Nowadays, it has become less. We have some protein related lab tests also, like liquid chromatography and also study that also. We have hemoglobin, electrophoresis, all those related questions also. So yeah, prepare well and we'll be seeing you tomorrow sharp at 10 p.m. So don't miss this.
Yes.
Once that is done, then we'll have a session on enzymes and then we'll have the grand finale.
And guys, we would like to let you know that we'll be hosting this grand finale very soon. And just for your own sake and your enthusiasm, we are going to keep the prize money to half of the number of participants. So I hope that you're very much eager to get validated and get these rewards in your name. Looking forward for you and for us. So keep studying and stay connected. And don't
Think
forget
tomorrow. Yeah,
there is a something known as cutthroat competition, and you got to be in it, okay?
yeah. So all the best for your preparation. We'll see you tomorrow night. Till then, goodbye.